Okay, welcome back, everyone. My name is José Luis Marjan. I'm a faculty member here at the Institute and had asked me to chair this session for one of the missing speakers that was supposed to do so. So I'm happy to help out. First speaker this afternoon is Marjan Huizing from the National Human Genome Institute at NIH. And she's going to be talking about disorders of sialic acid synthesis, pathways, and prospects for therapy. Marjan?
Okay, good afternoon, everyone. I'll start with the simple pathway information as everyone comes in, and I'll explain the more harder things later on when everyone is here. So thank you, Hud, for the kind invitation to present our work on sialic acid synthesis pathway and prospects for therapy in this exciting meeting. I have to disclose that for the work that I present, we receive support through a CRADA with Escala Therapeutics for the clinical development of ManNAc. Sialic acids, as you probably all know, are negatively charged acidic sugars that sit as terminal residues on end groups of glycans, glycoproteins, and glycolipids that mostly cover membrane surfaces. I don't know, do we have a pointer? Okay, well, you see the, does it work? Oh. Okay, huh? Oh.
The first, it's right here, this red thing.
There you go. There you go.
Okay, there. All right, so glycans mostly sit in membranes, with their glycan structures kind of pointing out, covering the membrane, and these red residues here are the sialic acid residues. So you can kind of imagine the sialic acid residues covering the membrane, playing many important roles there, in both in normal physiology, like many signaling functions, or in pathological processes as well, such as microbe binding or immune response. Sialic acid is produced in the cytoplasm of cells. And the first committed step to make sialic acid is when UDP-glucNAc gets converted to ManNAc and ManNAc 6-phosphate, which is mediated by a bifunctional enzyme, UDP-glucNAc 2 epimerase, ManNAc kinase, or abbreviated GNE. Then two more steps will make it sialic acid, which goes to the nucleus to become activated to CMP sialic acid, which can be used by the Golgi to sialylate glycans. Cytoplasmic CMP sialic acid also feedback inhibits this epimerase enzymatic activity in the allosteric site, thereby creating a tight regulation of the amount of negatively charged sialic acid present in the cell. As far as I know, there are 3 human genetic disorders associated with steps in this pathway. The first one is CDG type 2F. There are only 2 patients so far described with this disorder. Patients have mutations in the CMP sialic acid transporter in the Golgi. There is no therapy available. The second disorder is sialuria. 7 patients in the world described so far, which is caused by a dominant, one allele, missense mutation in the allosteric site for CMP sialic acid binding in the GNE protein. These patients constitutively make free sialic acid in the cytoplasm and excrete gram quantities of sialic acid in their urine. No therapy available. Then the third disorder is GNE Myopathy, of which I spent the majority of my talk today. It's caused by mutations in GNE, either the epimerase or the kinase domain encoding regions. These patients have decreased enzyme activities, decreased amount of sialic acid produced, and presumably reduced sialylation of glycans. GNE myopathy has many historical names. Shown there. It's caused, as I said, by biallelic, mostly missense mutations in the GNE gene. It's a rare young adult onset progressive disorder, which initially present with distal muscle weakness, mostly foot drop, eventually progresses to severe weakness and atrophy of all muscle groups, but the quadriceps remain relatively spared, mostly in the early onset of the disease. Shown here in an MRI, in black you see the quadriceps muscle relatively spared, but in later stages of the disease that becomes affected too. Within 10 to 15 years after onset, most patients become wheelchair bound and are depending on dependent care. The exact pathological mechanism of GNE myopathy remains unknown, even though the gene was identified 15 years ago, but is most likely due to hyposialylation, and therefore there are good prospects for sialylation-increasing therapies. So we found with lectin staining that muscle tissue of GNE myopathy patients is hyposialylated. Shown here for 2 lectins that I will show other results on throughout my talk. So the SNA lectin binds to, lectins are proteins that bind to end group monosaccharides on glycans. The SNA lectin specifically binds end group sialic acids in a 2,6 linkage to their underlying monosaccharides. So normal tissue has a bright stain or signal for SNA, indicating full sialylation. And we use VVA that stains, that recognizes O-linked GalNAc on O-linked glycans with the sialic acid groups off. So it indicates there's desialylation. In normals, VVA give no signal as expected, fully sialylated. But 2 patients with GNE myopathy muscle show desialylated, decreased SNA signal, increased VVA signal. Oh, yeah, so SNA here, I pulled that cartoon back up. You can imagine that it covers the membrane and shows this SNA signal there. 4 sialylation-increasing therapies have been are under evaluation for GNE myopathy models and humans. We have put a lot of effort in, and we believe in the strength of using ManNAc and acetylmanosamine as a sialylation-increasing therapy. ManNAc is small, it is uncharged, it readily enters cells, likely by diffusion. It is historically used in cell culture systems to sialylate cells, to fully sialylate cells. And patients with, even with the kinase domain defect, can still make ManNAc 6-phosphate if you give ManNAc, because there are other kinases in the cell, such as GlucNAc kinase, that can phosphorylate ManNAc. It also feeds in after this feedback inhibited step. So you can continuously make sialic acid if you feed ManNAc. Other therapies that are being pursued is free sialic acid therapy, which may be a little cumbersome to get a negatively charged sialic acid into the cell. Highly sialylated glycans have been evaluated, such as sialylactose and IVIG, of which their sialic acid groups can be reused to sialylate other glycans that needed in this disease. We have performed a clinical trial, a pilot trial with 4 patients with GNE myopathy, giving them IVIG, and the results were inconclusive but promising. And gene therapy has been pursued in 1 patient where the GNE gene was injected both IV and intramuscular, and those results are also inconclusive. Okay, so preclinical models, like animal models of GNE myopathy, have shown good prospects for ManNAc therapy. We studied the M712T knock-in mouse model, mimicking a Persian Jewish founder mutation for this disease. And to our surprise, the mutant mice died before 3 days of life of severe kidney disease, glomerular disease. But when we fed the pregnant female mothers with ManNAc in their drinking water, 50% of the mutants survived. It rescued their kidney phenotype, and they went on to live into adulthood without further ManNAc supplementation, and they developed an adult onset desialylation of muscle tissue, similar as the human patients. So it's again the SNA and VVA, normal, fully sialylated. The mutant mice muscle is desialylated, and then we gave ManNAc therapy for 12 weeks to these mutant mice, and we showed full resialylation of muscle tissue with ManNAc, oral ManNAc. Okay. Another group mimic in a transgenic model for this disease mimicked a common Japanese founder mutation, where the mouse model recapitulated the human model a little better in that it did not die of early glomerular disease, although the kidneys were somewhat affected. In these mice, strength studies were done. And as you see, the mutant mice had decreased muscle contraction and decreased motor performance, but after ManNAc therapy, did this increase to almost a normal range. So these and additional preclinical data have triggered the start of 3 clinical studies at NIH. All 3 studies were run by Dr. Carrillo as a PI, and for ManNAc and GNE Myopathy, Dr. Gall is the IND holder. We got great help from the Therapies for Rare and Neglected Diseases program, as you heard from Dr. Tabak this morning, in acquiring an IND for treatment of this disease. A little bit about all 3 studies. The natural history study for GNE myopathy patients started in 2011. 44 patients have been enrolled. It's a longitudinal prospective single-center study, and patients come for follow-up visits to the NIH Clinical Center. And biochemical and clinical parameters are assessed to identify biomarkers, both for diagnosis and for possible response, for use to possible response to therapy. It's very important to find a good marker for an early diagnosis. Most patients have an about 10 years diagnostic delay, and these may be the 10 years that they're most responsive to therapy, so it's important to identify them early. Clinical parameters are used to determine disease progression and to design a treatment trial. It can be used for estimating the length of the trial, the amount of patients needed, frequency of assessments, and importantly, our natural history study data has aided the design of a very novel muscle progression model that is now being evaluated to use for clinical endpoints in this slowly progressive muscle disease. The phase 1 study of ManNAc is completed. It was a first-in-human study. It was placebo-controlled, double-blind, single-dose. 22 patients in 3 cohorts were evaluated in a single dose of ManNAc and 2 placebos per cohort. The results, the ManNAc was safe and well tolerated. There were no serious adverse events. And the ManNAc PK, which I will show, looked very favorable for only twice a day oral dosing to continue to result in continued free sialic acid production. So this is the ManNAc PK, plasma PK in the 3 cohorts that we found. 3 grams, 6 grams, 10 grams. So ManNAc peaks in plasma at 4 to 6 hours after taking a single dose, and it's out of plasma within 10 to 12 hours after dosing. Of course, we were interested in how, if you feed ManNAc, how that would increase free sialic acid in plasma. So this is the background level of free sialic acid in plasma. And this were the free sialic acid levels after 3 gram, 6 gram, and 10 gram of a single dose of ManNAc. So as you see, free sialic acid peaks 10 to 12 hours after taking a single dose of ManNAc. The free sialic acid importantly in the higher doses, remains increased even 48 hours after taking a single dose of ManNAc. Free sialic acid levels seem to not increase above a certain threshold. So it does not matter if you give 6 grams of ManNAc or 10 grams of ManNAc, you cannot up the free sialic acid levels more, introducing a very nice safety mechanism for not getting to sialic acid toxicity. So based on these results, it was decided that ManNAc could be dosed twice a day, because every time you give a new dose, the previous dose is out of the system, and you continuously increase free sialic acid levels. However, they will not reach beyond a certain threshold. This was used for a ManNAc Phase 2 trial in patients with GNE myopathy. They received 6 grams twice a day, oral, dissolved in water. 12 patients are being dosed. They have all completed 90 days with a muscle biopsy before and after 90 days. And they're continuing into a 1-year study. Multiple dose ManNAc was safe and well tolerated. There were minor adverse events, mostly GI related, bloating or loose stools, because you have a large amount of sugar in your system. The plasma PK follows indeed that we did not build up ManNAc in plasma, and you continuously you increase sialic acid to a certain threshold level. We're studying efficacy, of course, clinical and biochemical parameters. Strength is being measured and being validated in the disease progression model. We test MRI, patient-related outcomes. We test plasma and intracellular markers, including CMP sialic acid. And we have muscle biopsy tissue to evaluate for resialylation. These data are still under evaluation, which I cannot present today. Now, it looks like ManNAc is a very good compound to increase sialic acid in humans and safe. So would there be other disorders of hyposialylation that could potentially benefit from ManNAc therapy? So remember that our mouse model had glomerular disease, and it could be completely rescued by giving ManNAc. So we decided to further evaluate the kidney disease in our mouse model. There's not enough time to go into the very details, but after ManNAc therapy, we decreased proteinuria, improved kidney histology, and improved the sialylation of glomerular glycans, which we showed with a lectin staining panel. We used 2 lectins that bind end group sialic acids, WGA and SNA, present in normal, absent or highly decreased in mutants, which restored after ManNAc therapy. And all these lectins only bind to glycans when the sialic acids are off. So you see them all increased in mutant mice and decreased in treated mice. So these and other, let me see. Oh, no. So we picked 2 lectins, 2 informative lectins to test human kidney biopsies, just to see what sialylation looks like. So first we acquired 40 biopsies of different glomerular diseases, different diseases across the board, and we stained them with SNA and HPA. And to our surprise, in 8 patients, we found severe hyposialylation, and decreased sialylation was never recognized or described in glomerular disease. So most patients showed a normal pattern with a lot of SNA staining present, but these 8 patients had absent SNA and highly increased HPA staining. Since we received these kidneys de-identified, we could not go back and correlate them to clinical parameters like steroid resistance or proteinuria levels. So for that, we started a new collaboration with the Neptune Consortium, a nephrotic syndrome study network, from which eventually we got 91 kidney biopsies from patients with the 3 primary glomerular disorders: focal segmental glomerulosclerosis, minimal change disease, and membranous nephropathy. And to our and everybody's surprise, we found 70% of these samples to be hyposialylated with our two lectins. So we are now, we have received all the clinical data of these 91 patients, and we're doing statistical analysis to see if it would if hyposialylation correlates to some of these clinical features. So these preclinical data even resulted in writing an IND. Even though the exact underlying mechanism of this desylation remains unknown, the preclinical evidence was good, and our experience with ManNAc and our animal toxicity data on ManNAc could be repurposed from our GNE myopathy trial. And on top of that, there are very few therapeutic options available for patients with glomerular disease. So this was a very well-received therapy. An IND was approved in 2015, and the clinical protocol has been IRB approved by the NIDDK IRB in January, and the study is currently recruiting. Dr. Gall is the IND holder, and Dr. Kopp from NIDDK runs the trial. So now what about are there any other disorders that have hyposialylated tissues? So there are some indications in very recent literature. For example, in a mouse model, they showed that ManNAc therapy can improve sleep-wake quality. And another mouse model of rheumatoid arthritis got benefited from ManNAc therapy, in that ManNAc resialylated IgG sialylation and improved the phenotype. So these are very exciting data, and it's a very exciting time to further develop ManNAc as a therapy for these hyposialylation disorders. And our lectin stains can definitely help identify such disorders. ManNAc therapy, we're very willing to collaborate, to use our preclinical data to help with the lectin stain, to promote ManNAc therapy, to increase sialylation. So we're open for collaborations. So this whole project is a multi-institute, multi-year effort at NIH, many institutes involved, extramural collaborators, industry collaborators, some key persons, Dr. Gall and Dr. Gall's lab, Dr. Carrillo from NCATS, Dr. Kopp from NIDDK, and of course all the patients that participate in the study. Without them, we could have never come this far. Thank you.
Time for some questions?
Hold on.
So why do you think muscle and kidney are so affected and other tissues aren't? And I guess the second part of that is, it looks like O-linked glycans are the ones that are hyposialylated.
Yeah, that's right. So first, why kidney and muscle? So we hypothesized that if you run out of so the patients have GNE deficiency, if we go for GNE myopathy. So you are born or you live with a certain amount of sialic acid, and you can produce some in the pathway. Oh, and you can produce some in the pathway. So during life, you may get a deficiency, a deficit of the amount of free sialic acid available to sialylate your glycans. And the first ones that get affected may be sialic acids for which sialyl transferases have a low affinity. Like it may be that the 2,6-linked sialyl transferases that mostly sialylate O-linked glycans have a lower affinity for CMP sialic acid that runs out. So all the N-linked glycans will be fully sialylated. Transferrin is normal in this disease, but the O-linked glycans may lag behind. So muscle, like alpha-dystroglycan, has a lot of O-linked glycans, and we found hyposialylation of those proteins. So that may be one explanation. Why muscle first? Why kidney? We only found kidney in the GNE model in mice. So there we can explain it in a similar fashion, that you may run out of free sialic acid available. Why it is in the human glomerular diseases, it's clearly a secondary effect. Because we find it across the board in different glomerular diseases, so it cannot be a genetic mutation. It is a secondary effect resulting from primary injury that results in desialylation. We have some indications, but yeah.
So you mentioned alpha dystroglycan. Have y'all looked with like 2H6 antibodies? Is alpha dystroglycan glycosylation?
So we have stained with the 2H6 and the 4, yes. So we only used this in 4 patients. In 2004, we found all 4 patients to be desialylated in alpha dystroglycan, while the core protein was there. However, in the literature, other groups have done the same. They find some patients do have sialylation of alpha dystroglycan. So it is not 100% clear.
You use the 2H6 antibody,
Yes.
which is sialic acid independent.
Yes, and yes, that's so, and the core glycan. Yes, so we published it in 2004. And, yeah.
Okay. And along those same lines, the transporter patient, the patient that's got to meet
CG2F.
Yes. So is there alpha dystroglycan?
So I don't know. So the first patient died at 37 months, and the second patient was just described by Eva. So I think the second patient had more severe symptoms, renal and cardiac symptoms, seizures. So there may be hyposialylation.
But we never.
Yeah, I know. Yeah.
So he died before we actually. Thank you. So it was an adult with congenital abnormalities like a cardiac, like an aortic valve abnormality. There was significant glomerular disease.
Yeah,
so
I
it's
know. Yeah.
really
And
very possible.
so I would be interested to see what the third patient that has been identified, what features that patient have, and if kidney biopsies are available. And maybe just a preventive ManNAc therapy can be tried if there is no other therapy available.
We'll check. Just a quick question. Have you ever seen altered sialylation or glycosylation in tubular illnesses?
Over-sialylation? Yeah.
Under
Under-sialylation in what?
renal tubulopathies?
So that's interesting. With the lectins that we use are mostly focused on O-linked glycosylation. We do not see any tubular defects. We see full sialylation, like with the WGA lectin of tubules. So it looks like tubules may have an abundance of N-linked glycans that are not affected in these diseases that we study. It's very possible that tubular diseases have desialylation, but we did not look at them. We didn't look for them. We look at glomerular disease.
All right. Thank you very much.
Yeah.
So our next speaker is Raymond Wang from Children's Hospital of OC is Orange County, that's correct. Talking about mitochondrial GFM1 deficiency presenting as a congenital disorder of glycosylation. Okay.
All right. Good afternoon. Just wanted to thank HUD and Bobby for inviting me here. Grateful for SBP for having me here to talk about this, which is a little bit off the beaten path from the standard CDG world. So, whoa, how do I do this? Oh, please, this. So just some brief introductions. I am a biochemical geneticist at the Children's Hospital of Orange County, which is in California, not too far from here. Been there since 2007. I have been a long-time friend and collaborator with HUD and Bobby ever since we came to Orange County. Basically, any case that has an abnormal glycan pattern, I send them an email and say, help me, please. And actually, right now, we care for a small number of patients with CDGs, some of them who may be here this weekend. So thank you. So as with any of these rare disorder cases, it always starts off with a good story. So many, many years ago, when I basically first started as a physician, as a clinician at Orange County, I was asked to see a 3-month-old little boy with RSV infection. And I have to admit that my first reaction then was, why are people asking me to see a baby that has a respiratory infection? But, you know, when you take the time to delve into the story a little bit more, the mother told me that she was sure that something was not right with her child from the very beginning. And I know that that's a story that's first sentence that's shared by a lot of patients. Essentially, he wasn't really looking at her. He was not fixing on faces. He was not smiling at her. He had microcephaly, or small head size. And I think what was more significant and really got my attention was that she had told me a story of her older son, who had very, very similar symptoms to start off with, had microcephaly and delayed development, but also ended up passing away at 15 months of age with essentially what you could call multi-organ system failure. He had cardiomyopathy, or enlarged heart, seizure disorder, and ultimately it was the liver failure that probably caused the passing of her older son. And so when I went and saw him, he also had microcephaly as well. His head size at 3 months of age was basically the same as a newborn. He definitely didn't look at me, didn't smile. He had poor head control, and his arms and legs were stiff, so he had hypertonia. Given these findings, as well as the family history of serious early onset disease and the lack of a prior workup, they had come in, she had wanted to see a geneticist or a metabolic physician for quite a long time. Essentially, the physicians wanted to facilitate the metabolic genetic workup there while he was hospitalized with the respiratory infection. So we requested a number of tests to screen for a large number of different conditions. One of them included an MRI, which at the time showed a normal brain anatomy. We also looked at his eyes to look into why potentially he wasn't paying attention or fixing on faces, and at least the anatomy of the eye was normal. Now, one thing that we did see that was very clear was that his lactic acid was elevated, and it was consistently so. Lactic acid is always somewhat problematic because drawing blood from a small infant may not be the easiest thing, and so whenever you have a baby that's crying, a baby that's struggling, a phlebotomist that's having trouble finding a vein, you can get elevated lactic acid. But his was very consistently and very highly elevated, which is beyond what you would see, from a blood draw difficulty. And when all the results came back, there really weren't any abnormal biochemical findings, with the exception of carbohydrate deficient transferins, and that's why I'm here today. So at the time, Mayo Clinic was running those tests for us, and he had a type 1 pattern. And so the mono-oligo to di-oligo ratio was elevated. If I remember correctly, it's like 0.16, and their normal at the time is 0.1 or below. And so it was a little bit unusual, right? Because in CDGs, you read about patients with cerebellar hypoplasia or seizure disorder, abnormal nipples, or fat distributions. At least that was the case in 2010. And so I repeated them, and they were abnormal again. And since I didn't believe that, I repeated it for a third time. And when they came back abnormal for the third time, that's when I got a little bit of extra testing, and I contacted HUD and Bobby. This weird little tetrasaccharide came out on a glycan analysis that was run by the folks at Emory at the time, and ultimately I was reassured that there really wasn't a whole lot of significance to it, but I throw that out there in case somebody recognizes it for any potential significance. So since this patient didn't really fit any existing pattern for CDGs, I pulled up the bat phone and I contacted Bobby and HUD. And this actually comes from the Mite Family's website, so with credit to them. And we were able to obtain DNA from the living family members. But more importantly, we had discovered that the older brother had had fibroblast analysis for pyruvate dehydrogenase deficiency, which also causes elevated lactic acid. And the lab that ran the test actually kept his fibroblasts frozen. So we're actually able to obtain DNA from his deceased sibling. Meanwhile, as the lab research testing was going on, our patient was following a very similar pattern to what was going on with his older brother. And that was definitely causing a lot of concern for the mother because she knew how that story went. He started to have seizures at 4.5 months that were actually specifically infantile spasms. He had severe feeding problems and trouble gaining weight, so eventually a feeding tube was placed. And he developed a renal tubular acidosis, for which he required bicarbonate supplementation. And about 8 months is when she really got concerned because his liver started to get enlarged. He had increased liver enzymes, and he had a bleeding diathesis. He would become hospitalized roughly once a month, and sometimes even more frequently with routine illnesses, so a fever, a flu, vomiting, and diarrhea would actually cause him to have very, very severe acidosis. Sometimes his bicarbonate level would be unmeasurable by our laboratory. His blood pH was anywhere from 7 to 7.1, which is very, very acidotic. His lactic acid was elevated every single time, low blood sugar. And what we discovered, sometimes you don't necessarily need to know exactly what the patient has, but you know how to treat them. And so we discovered really, really quickly that a high glucose infusion rate, giving him more sugar than the average dehydrated patient, would fix not only the acidosis, but also the lactic acid and the low blood sugar. And so we got really quickly the hang of how to take care of him. And he would stay hospitalized for a few days and then go home. But this would happen recurrently every month. So I actually went back to my emails, and I'm quoting what folks, me and HUD and Bobby had written, but I wrote, I have yet another puzzler. And this is in July of 2010 when we had the CDG reports. A few months later, because of what was going on clinically, just some correspondence back and forth, I wrote, he is behaving much like a mitochondrial electron transport chain deficient child with the lactic acidosis. And that was just musing more, but I had no idea how prescient that would be. Almost 2 years later, Bobby sent us an email that said, I think we figured out your case. And I just want to contrast that for the families with the Human Genome Project, which was started in 1990 and not completed until 13 years later. I think my point for this slide is not to complain about how old or how long it took to get an answer, but to just really draw a contrast between the time that's elapsed now between identification of a case versus actual identification of a diagnosis. At the time, in 2010, there were no labs, commercial labs at least, that did exome testing. And if you could only sequence one gene at a time in a commercial lab, often to the tune of $2,000 to $3,000 per gene, and exome testing was really only done on a research basis. And for families who have gone through the research process, we understand a lot of the time it's just like sitting there waiting for the toast to pop, and then suddenly when it actually comes out, you're like surprised with the answer. And so that's kind of what we felt like. It was really, really rewarding to get that email that finally potentially an answer had been discovered. So one more time. Contrast this with now, where commercial whole exome testing is available, where more than 20,000 genes can be sequenced in 2 months for, retail cost of perhaps 7,000. So what did we find? What did Bobby find more specifically? There's a gene called mitochondrial GFM1, and in this family, 2 genetic changes were found, one from each patient. So both parents are carriers. We call this biallelic biparental inheritance. This change, which is an arginine to tryptophan substitution at 250, was found in the mom, and that actually had been previously reported in other GFM1 patients, which is very encouraging to us. We didn't have to go through a long process, at least for that mutation, of demonstrating whether or not that was disease-causing. However, in the father, this intronic change was discovered, and that, as you can tell, this number 998 is not what you usually see for a splice site mutation. It's usually a much smaller number, and that was an impressive piece of work that Hud and Bobby's lab did to demonstrate that, in fact, this was actually pathogenic, and we'll go through a little bit more detail of this, but we were able to find that the patient had both mutations. His deceased brother had those 2 mutations, and his healthy sister did not. And so the presence of these changes segregated or correlated with the presence of the disease. Now, as I was putting together this talk, I got nervous because I realized that families would be here, and so I put in a bit of a genetics primer, and I apologize if it's a bit basic for the researchers here to go through the central dogma all over again. But for the family's sake, go through a few slides of this and bear with me. And so all the information that's needed to make and maintain each one of us is located in what's called a genome. And our genome is composed of deoxyribonucleic acid, DNA, which is organized into genes. And genes are like individual recipes within cookbooks. And so they tell the cells of the body how to make things, make proteins that, do important functions within the cell. Now there's a step in between the genes and our proteins, which involves something called RNA. And that's basically read and copied by our cellular mechanisms, enzymes, in a process that's called transcription. And then RNA is then read and turned into a protein. However, it's a little bit more complicated than that. So not all of our DNA that we have in our cells actually encode a recipe. And so exons are the portion of the DNA that actually code for things. Everything in between is called an intron. And so you can see here, all these dark blue things are the exons, and everything in between are introns. And what happens as an mRNA is made is that the cells actually isolate or splice out and get rid of the introns and put everything together to form a mature RNA, which is then read into a protein. Now, you've heard of whole exome sequencing and whole genome sequencing. Basically, whole genome sequencing is where every single letter within the genome is sequenced. And right now, because there's so much information in that's definitely not clinically available, and I would say it's rarely done on a research basis. Whole exome sequencing, however, is where you sequence all the coding regions, the parts that actually turn into proteins, and that's what we've been talking about here. 2 more slides, bear with me. So mitochondria are little things that live within most of our cells, and they make energy that power all of our cells' functions. And so here's a little mitochondrion. When we zoom in, you'll see that there's a couple of membranes, and you'll see loops of DNA. And what you see as ATP synthase. And that's an important part of the machinery that actually converts things called electron carriers into energy, ATP. So we zoom in on that a little bit further. The things that actually make energy inside of our mitochondria are called complexes. We have 5 complexes, and essentially what happens is these electron carriers get passed through each of these complexes in order to make ATP. Complexes are made of proteins. And what's important about mitochondria is that while the majority of these complexes, proteins, are made by the DNA, the genes in our nucleus, there's a few complex subunits, for example, 7 in complex 1, 1 in complex 3, a couple in 4 and 5, that are encoded by the DNA of the mitochondria. So the mitochondria itself has a separate genome that's distinct from the nuclear genome. And all of those combine and work together to form the complexes that power our cells, power us. So the same process of reading DNA and turning it into RNA and into protein actually goes on within our mitochondria as well. It's a process that's distinct from what goes on outside of the mitochondria and what's called the cytoplasm. And so when you cannot turn DNA into RNA and then into protein, what ends up happening in the mitochondria is all of those complexes that are encoded by mitochondrial DNA, so 1, 3, 4, and 5, they're not as functional. They don't have the right units. And so, well, they're sick, okay? So your mitochondria are sick and they're not able to, or they're less efficient at generating the energy that your body needs. And if you think about it, obviously all of our cells require energy, but some cells need more energy than others. So your brain cells are very, very metabolically active. Your heart cells are metabolically active because they're beating, once a second, sometimes even faster. Your liver cells are very metabolically active because they're conducting many, many different reactions that are important. Kidney cells. And if you think about it, those are the symptoms that were affecting, this family: seizures, cardiomyopathy, liver failure, and renal tubular acidosis. So it fits. So perhaps we have a diagnosis, and if you actually look in OMIM, which is the portion of the NIH website that lists genes and disorders, GFM1 deficiency is called combined oxidative phosphorylation deficiency. And then when you look at the alternative names, it's like hepatoencephalopathy, early, fatal, and progressive. And so when you read the descriptions of these patients, these are kids that are passing away very, very early on in life, which may be what went on with the older sibling, but not with ours. So here's where we collaborated with a number of folks all across the country to establish that this family's genetic changes actually resulted in biochemical deficiencies. Okay, so let's skip that. So we need to first demonstrate that those mutations actually result in abnormal mRNA. And Bobby was able to demonstrate that the father's mutation actually causes what's called abnormal splicing. It actually creates a splice site for a cryptic or a hidden exon that's not usually found in the normal gene. And so essentially an extra chunk of DNA is read in and encodes an extra portion of RNA, which usually would not turn into a functional protein. And indeed, that's what we saw. So the patient and his brother have an RNA section that is of increased length compared to his parents, which is exactly what you'd expect with an extra exon that's been spliced in. Further, we found that the patient and his brother's fibroblasts made hardly any GFM1 protein in their, and so you can see there's hardly any GFM1 here compared to a parent. And then finally, we were able to correlate, and this is with help from the University of Colorado in Denver, that our patients had markedly reduced complex 4. So this brown stain here is what complex 4 should be, and patient 1 and 2 had very, very little complex 4. And complex 5 was not assembled properly. And so complex 5 usually should be one band, and you see multiple little bands here, which imply that the complex never assembled properly and therefore doesn't work. So a little bit more about GFM1 deficiency in the time that I have left. It was first identified in 2001. The gene itself was not identified until about 15 years ago. It was first linked to human disease in 2004, with 2 siblings having lactic acidosis, severe liver failure, increased muscle tone, and early death. 2 additional patients were discovered in 2006 with very similar manifestations. And so far, there have only been 12 patients. So this is a rare disorder for sure. All but 1 of the patients have had very similar symptoms: lactic acidosis, liver failure, seizures, and death in infancy, with the longest surviving patient at 2 years. There's 1 patient recently reported who's still alive with some issues, but appears not to be progressive. Here's a slide that summarizes all 12 patients, which for the sake of time I won't go through, aside from the fact that they have features of mitochondrial abnormalities and early death. Okay, big question. How does GFM1 deficiency actually result in CDG? And the truth is we don't know. We've asked to go through the database and see if there are other patients identified having mitochondrial disorders that result in abnormal glycosylation. And we may have gotten a lead, but then couldn't get any more clinical information on that family who had MPV17 deficiency, which is another mitochondrial DNA depletion syndrome. There was 1 report of a patient initially thought to have mitochondrial disease because of the multisystemic symptoms, but then eventually identified as having PMM2 CDG. And I have to say, transferrins are not routinely checked in patients that are being worked up for evaluating suspected mitochondrial disorder. So this may be just a fact that we're not looking. It might be out there, but we're not looking. We think that perhaps when there's liver synthetic dysfunction, the glycosylation that goes on is not normal. And in fact, the patient has what's been called von Willebrand's disease, which is a bleeding problem, which is seen very frequently in other CDGs because, you know, their clotting factors are glycosylated. And the exome didn't actually identify any genes changed in the genes that cause von Willebrand's. Are there any treatments? There's nothing curative at the moment. There was 1 report that did talk about gene therapy, which was able to rescue fibroblasts, skin cells, from a patient who had GFM1. We had the patient on levocarnitine and coenzyme Q10, which are standard supplements that are given for mitochondrial patients. How is he now? He just turned 6, actually not too long ago, so he may be one of the longest surviving patients. Did we do it? I'm not sure. I hope so. I'd like to claim credit that maybe, you know, some of the care that we did has enabled him to outlive his older brother. He currently has no seizures. They're very well controlled on 1 medication. He has delays, but he smiles all the time. He is the happiest little boy. He is sitting and crawling and trying to walk, which I think is more than any of the kids had ever tried before. He's now hospitalized maybe 3 or 4 times a year, and his liver disease is actually thankfully stabilized. We at HUD and Bobby's request obtained transferrins just a few months ago, and they're completely normal. And I don't know how to explain this. Perhaps the population of cells that was not working well in the liver has been selected out or something. I have no explanation for that, but I do know that this has been reported before in other CDGs. So, in conclusion, rare diseases are everywhere. They are not so rare. You just need to look. Different genetic or metabolic conditions may mimic one another. And so we had thought that this patient had a CDG and in fact turned out to have mitochondrial disorder. I had cited a paper where people thought that the patient had a mitochondrial disorder and ended up having a CDG. So we need to look. And I think I just wanted to highlight the dedication and perseverance of actually not just the physicians and scientists, but also this family who kind of, labored with us through all the hospitalizations to find an answer for this family's condition. So we are hopeful this will result in deeper understanding of mitochondrial function as well as development of other treatments. So thank you.
We have time for just one question. Go ahead.
Yeah, actually more of a comment than anything else. So this is a place where we really need to be talking to our mitochondrial colleagues, because last year's UMDF meeting, do I really need a mic?
Probably not.
Last year's UMDF meeting had a whole session on the glycosylation of the mitochondrial proteins, and that they have to be glycosylated to get through all the membranes. And when you start to really look at a number of CDGs, you will find amino acidurias, white matter disease, generalized atrophy, lactic acidosis, although not that high, CKs. Liver disease is a little different in most CDGs. The liver disease is severe early on and gets better over time. So I think that we have significant muddy water between CDGs and mitochondrial disease. And everybody that hangs out with me does look for CDGs when we think mitochondrial disease, because we know those proteins are highly glycosylated.
And many of the proteins you're talking about that go from mitochondria are O-glucanac and not the N-link variety. So it looks like there could be some mix-up, but maybe you get a window like we're seeing here. Maybe that's part of it.
We've gone through a lot of different types of glycosylation today. Today, what I'm going to be focusing on is specifically O-Mannosylation. And the reason we focus on O-Mannosylation is that's the type of mannosylation that functionally modifies alpha-dystroglycan so that alpha-dystroglycan can interact with the extracellular matrix. I wanted to start my talk off with acknowledging the people who actually do the work, even though when I grabbed this picture a couple of days ago, I realized the two main people that did the work that I'm going to talk about are not in this picture. This is some of our group. And then also just wanted to show the CCRC. The CCRC is a somewhat unique place. There's 18 PI labs, about 240 people working every day, and the only thing we care about are carbohydrates, right? So it's a complex carbohydrate research center at the University of Georgia. And so I thought I'd start off a little bit, just presentation of disease. What do we see in the clinic when we're going to run into a case of Dystroglycanopathy? And usually, of course, it starts off with what's often referred to as a floppy baby. So a child presenting with hypotonia, failure to thrive, and their CK levels are tested, and they're usually very high, right? And CK is just a measure that probably indicates that there's muscle damage in these people. And so the next thing, unfortunately, is a pretty intrusive process, and that is you're usually going to get a muscle biopsy. And I'm not going to talk about it. I talked about it 3 years ago. We've actually now developed a blood test instead of doing a muscle biopsy here, which would be much better for the patients because oftentimes these children are severely affected and they don't really understand why you're hurting them. And so, but with a muscle biopsy, what we won't have in a blood test is you actually get the muscle histology and immunohistochemistry. And of course, these days we do gene sequencing. And really, this is what you would consider a normal looking muscle. This is a very abnormal muscle. This is a patient that actually is a Walker-Warburg syndrome patient. And this is just an H&E stain of that muscle. And really the key for us are these two antibodies, 2H6 and VIA4. And these two antibodies do not recognize the protein. They recognize a repeating disaccharide on the protein alpha-dystroglycan. And so what they really recognize is functional alpha-dystroglycan. And so if we look with these two antibodies in a patient with Walker-Warburg syndrome, we basically get no staining at all, right? And so we know exactly, at least partially, what's going on now. And so with congenital muscular dystrophy as a very large family, right? The key word being congenital, so the symptoms usually present at birth. You have hypotonia, muscle weakness, contractures. Motor developmental delays are very common, and some of the neurological impairments, unfortunately, are quite common and can be quite severe. And what I'm going to focus in on, what my laboratory studies, is the so-called dystroglycanopathies. And so the dystroglycanopathies also range in severity quite a bit. You have people with relatively normal lifespan that have mild limb-girdle disease, so they might go into a wheelchair in their 30s or 40s, all the way to Walker-Warburg syndrome, or unfortunately the patient usually will pass away in the first 18 months of life, and they have severe skeletal cardiac muscle defects, severe intellectual deficiency. So there's a really wide range of diseases associated with the dystroglycanopathies. So what's going on here? Because if we actually look at alpha-dystroglycan, it's called a dystroglycanopathy, right? There's only one family that I'm aware of in the whole world that has a mutation in alpha-dystroglycan. The mutations in all these dystroglycanopathy patients are actually in glycosyl transferases that add sugars onto alpha-dystroglycan. Alpha-dystroglycan's job in this complex is basically to hook up, inside your cell, you have a skeleton inside that cell called a cytoskeleton, and dystrophin binds that, comes up through the plasma membrane, interacting with alpha-dystroglycan, which then binds the extracellular matrix, that is another skeleton, if you will, outside of the cell. And so this whole complex's job is to hook one skeleton inside the plasma membrane to another skeleton outside that plasma membrane. And what's going on is we have a defect in glycosylation, because this isn't a protein-protein interaction. It's actually these LG domains that are on proteins like laminin, perlecan, agrin. They actually bind this repeating disaccharide sugar on alpha-dystroglycan. And so this is actually a patient we just recently characterized that has a mutation in a protein called TMEM5. And what you would normally see if you use an antibody that recognizes alpha-dystroglycan, the protein, in a normal patient, this is a skeletal muscle, it runs around 180 kilodaltons, and it's a little bit of a smear, and it's a smear because it has heterogeneous glycosylation. But in the patient, you see this huge shift in molecular weight. More importantly, the glycospecific antibody only recognizes in the patient, in the control, not in the patient. And functionally, if the job of the glycoprotein is to bind laminin. If you do a laminin overlay, of course, the control is fine, but you see no binding with the patient. And so several of us in the field over a period of years have tried to break down what's going on with O-Mannosylation. And O-Mannosylation really comes in three flavors, if you will. You have what are called core M1, core M2, and core M3 structures. You'll notice that core M1 and M3 look almost identical, right? A green circle, which is mannose, and a blue square, which is an N-acetylglucosamine. But they're in different linkages. One's 2-linked and one's 4-linked. We're not going to talk about these the rest of the time because these modifications actually occur on a lot of proteins. M3 structures, as far as the field can tell, only occurs on one protein, and that protein is alpha-dystroglycan. Also, the functional repeating disaccharide, that is what laminin is binding, can only be built on the core M3 structure. And so about, 5-6 years ago now, a collaborator of mine, Kevin Campbell, and I came across a really interesting result. We very seldom see phosphorylation on sugars in mammals. It's very rare. But they had done an experiment where they realized they could incorporate radioactive phosphate onto alpha-dystroglycan. And they could not remove it treating with alkaline phosphatase. So it wasn't a terminal modification. It had something on the other side of that phosphate. And so what we did is we treated, if it was a phosphodiester linkage, the great thing about a phosphodiester linkage is you can treat that with aqueous HF and it will cleave that phosphodiester linkage. And so what we were able to do is find this M3 structure with this phosphate hanging on this mannose. And so that started giving us an idea, because before that, all of us were thinking it was these M1 structures that were important. And that actually made us go look for and find the enzyme that was adding this GLCNAC in a different linkage, right, in the beta-4 linkage, as well as the enzyme that was adding this GalNAc. And it turned out when we found those enzymes, we then went to our 65% of patients with unknown genetic etiology, and were able to account for a lot of them, right? And so that was great. So we actually could start tying things together. So the question is, what's over here, right? And so there's been many, many genes established. I don't expect you to read this list, where we actually know what these genes do, right? We know what core glycans they're involved in. And there are patients with mutations in every single one of these genes, right? With the exception being that FUT9, which I'm not sure how that made it on my table. And then we've got these genes encoding hypothesized O-mannose glycan synthesis. That is, we have patients with mutations in these genes, and those patients present with dystroglycanopathy, hypoglycosylation of alpha dystroglycan. And so what my lab and other labs have been doing the last couple of years is trying to figure out what are these enzymes doing. And so we focused in on one of these enzymes. My laboratory does a lot of mass spectrometry. We'd looked at a lot of glycan structures. And one of the things we realized is there's mutations in B3GNT1 in patients with congenital muscular dystrophy. And it had been implicated in cancer and arenavirus entry, which is also very common with the O-Mannosylation pathway. But we did not have any glycan structure that had a beta-3 linked GLCNAC. And so we wanted to study this enzyme and see what was going on. And so what we did is we actually expressed B3GNT1, but we also expressed B3GNT2. There's no known mutations in B3GNT2 associated with muscular dystrophy, and B3GNT2 had been extremely well characterized compared to B3GNT1 by Thierry Hennett's group. And so we first just tested, it's called a B3GNT1, so it should be able to take a LACNAC and add a radioactive GLCNAC onto that structure. And B3GNT2 does that just fine. The B3GNT1 doesn't do that at all. So it's looking like B3GNT1 is not a B3GNT1, and that it's just been misnamed. And so we did, I think Reed mentioned what we always do when we don't know what to do, and that is we do a blast search and say, what other protein looks like this protein? And when we did that, we actually found that it's set down very tightly with one domain of this enzyme called LARGE. And LARGE is the enzyme that adds this repeating disaccharide, right? And so, and part of that repeat, it adds a xylose, it also adds a GLIC-A, and where the homology was right where the GLIC-A was. And so we hypothesized, oh, this is gonna be a GLIC-A transferase, maybe. And so what we did is we took every single sugar we could get our hands on, and we looked for the ability to take UDP GLIC-A with this enzyme and transfer. And what we found is it would transfer both to alpha as well as beta xylose. As a positive control, we used LARGE, and this enzyme is actually much more active than LARGE. You'll notice that LARGE can only add to alpha xyl. It can't add to beta xyl, but this enzyme can add to either. It can add to alpha or beta. We also did a technique called nuclear magnetic resonance. And the only reason we do nuclear magnetic resonance really in glycobiology is to figure out linkage and anomeric configuration. And so I'm not going to walk you through what all these little spots on a 2D gel mean. But basically what it did is it confirmed that it was a beta 1,4 glucuronal transferase. So B3GNT1 quickly became B4GAT1, right? And what was really interesting about this enzyme is if we start off with alpha-zyl, it will add a GLIC-A and stop. If you throw in large instead, which has both alpha, they can add both, both GLIC-A and xylose, it will take off very slowly on alpha, and then eventually it will catch up. But if you have an underlying beta xylose, you must have both B4GAT1 and large together in order to get extension. And so what this made us realize is, okay, this guy's probably priming for large repeating disaccharide, but we must have a beta xylose underneath. Because this enzyme, when you knock it out, you get congenital muscular dystrophy. So we must have a beta xyl, that's why it's needed. So at that point, and this is basically early 2015 where we had this phosphotrisaccharide, we have X, which means, right, that's code in science terms for we have no idea, right? And then we had a beta xylose, even though we didn't know the enzyme that did this. We had B4GAT1, which put on this beta 4 GLIC-A, and this served as a primer for this enzyme to then take off. And it's actually this repeating disaccharide, this beta 3 alpha 3 repeating disaccharide, that is what's functional. That's what you must have in order to get LG domain proteins to bind. All right, so how are we going to get to X? So I'll remind you, 7 or 8 slides ago, I showed you the way we got the phosphotrisaccharide on the peptide was to do HF treatment. So we backed up and we said, wow, it took us 6 years to think about this, but what did we release, right? What was on the other side? So we pulled the protein down and we said, what's left in the soup, right? And so the way we did this is we overexpressed a very small part of alpha DG and we expressed it in a cell, HEC293F cells, which are non-adherent cells that have almost virtually undetectable large activity. They have just a little, but it's really, really low. And so, skip past that. What that allowed us to do is do the HF treatment and then say, what got released? And what got released, much to our surprise, was a ribitol, so which is basically a ribose that's been reduced to an alcohol, right? So it's not really a sugar, it's a reduced sugar. Hooked to a xylose and a GLIC-A. And we also saw a little bit of ribose hooked to a xylose, a GLIC-A, a xylose, a GLIC-A, right? The repeating disaccharide. And so this got us really excited that we had a big problem. And that is, of all we thought we knew all of our sugar nucleotides. Well, we don't make any ribitol, right? We don't have a sugar nucleotide that's a ribitol. But what we did have is we have this enzyme called ISPD that is deficient in a large number of Walker-Warburg syndrome patients. It's probably the most mutated gene of all the genes right now. And so ISPD looks like a nucleotidal transferase. That's kind of the family that it fits in if you do these blast searches. And so what we did is we purified ISPD and we fed it ribitol 5-phosphate. And sure enough, it'll make CDP ribitol, a new sugar nucleotide, reduced sugar nucleotide. But if you leave ISPD out, right, it doesn't make anything. And we'll also do this with ribose, interestingly. And so now we've got an enzyme and defects in this enzyme cause disease that makes the sugar nucleotide. So that was getting towards figuring out what's going on with the structure. The next thing we realized is if we have a reduced sugar in a linear form, we should, if that's in the structure and it's before the repeating disaccharide, we ought to be able to take the protein and do what's called mild periodate treatment. And all that's going to do, it's going to cleave, if you have a ribitol that's open, it's going to cleave it. And so you would lose the 2H6 reactivity if we were right. And so that's exactly what you see here, right? MOG treated, you see reactivity with the antibody. If you do the mild periodate treatment, you lose the reactivity. All right, so we've got a ribitol, and we know it's before the repeating disaccharide. The next thing we did is we basically did the experiment that I showed you with HF to get the phosphotrisaccharide, but this time instead of treating with HF, we did mild periodate and didn't wait 5 years, grabbed the protein, and looked to say what is on the protein now. And I'm not going to take you through all the mass spec. All I want to show you is we get heterogeneity because there's multiple other types of O-glycosylation on this peptide. But at this site, we see phosphotrisaccharide with a ribitol fragment. And that fragment can be either a 2-3 fragment, so it cleaved between positions 2 and 3, or between positions 3 and 4, right? And I'll point out we did this on a high mass accuracy instrument, right? We're talking about less than 10 parts per million, right? Extremely accurate mass spec. And we did fragmentation, which I'm not showing you. So seeing 2-3 fragmentation and 3-4 fragmentation means the way this ribitol must be in there is something must be hooked at the 1 position and at the 5 position. Because if anything was hooked at 2, 3, or 4, you wouldn't get the fragmentation. And so we also still wanted to know what was going on right here. We know this is, we're pretty sure this is ribitol now, but what enzyme's responsible for that? So again, we take those unknown proteins for which there are patient mutations. And one of them was called TMEM5. And so what we did is we tested it against multiple sugar nucleotides and found that TMEM5 will just hydrolyze UDP xylose, right? It'll hydrolyze it. Then what we did is we actually had a TMEM5 null line, right? So it's from a patient that doesn't make any appreciable TMEM5. And we put back in this small part of alpha DG. And then we took TMEM5 after we purified the alpha DG from the patient cell, and we added UDP xylose radioactive, and we added TMEM5, and we got transfer. And if we mutated the site that we know on alpha DG has the M3 structure, it is not a substrate, right? Furthermore, we took TMEM5, and interestingly, we had a lot of trouble with this enzyme. We couldn't get it to transfer to ribitol, radioactive UDP xyl, or in this case, glow assay UDP xyl. And it sure would not transfer to ribitol 5-phosphate basically at all. So what we actually tried is transferring it to the CDP ribitol, right? To something that has a phosphodiester linkage in it to try to reduce that phosphate charge, because we thought that was most likely our problem. And sure enough, it'll transfer just fine. And we used the other cytidine using sugar nucleotide CMP, sialic acid, or Nu5AC as our negative control there, right? And so now we know TMEM5 is most likely the xylosyl transferase. So the 3 enzymes that we know come after ribitol: TMEM5, B4GAT1, and large. And so what we did is we used a Cas9 mediated knockout, taking these human HAP1 cells and just knocking these 3 genes out. And what you'll see is that the core protein shifts down substantially. It doesn't react with 2H6 in any of the 3 knockouts. And then we took 3 different proteins that usually bind alpha dystroglycan quite well, and none of them will bind, right? And so what that allowed us to propose is the following structure, right? So we've now replaced X, we think, right? And so we've got this phospho-man trisaccharide that gives some enzymes. We don't know what those enzymes are yet, but we've really only got 2 enzymes left. So it's almost got to be this FKRP fukutin. Add this ribitol. That then gets extended by TMEM5 and B4GAT1. And that small disaccharide allows to serve as a primer for this enzyme large to extend that repeat. And so we think we've worked out pretty much the whole structure. And I'm actually glad that my talk was a little late, because at 2:58 today, this paper got accepted by Eli. So, which Bob can tell you, we were on a plane together. We got off the plane, and one of our competitors published this very similar structure in Cell yesterday. So we're close enough that both our publications will be okay. What's interesting, and I should point out, because their structure and our structure are not the same. There's also a paper under review right now in Nature Communications by a European group, which has our structure, because I know they have our structure. The structure in the Cell paper was from Japan, and their data and our data are actually consistent with one another. I want to point that out. What they show is a phosphorybatol off of this GalNAc. And then, so basically, instead of all of this being hooked to this mannose, they show it all being hooked to this GalNAc, also through a phosphodiester, and they suggest that that phosphate is free. We'll see who's right? I mean, I don't know who's right? I mean, it's absolutely, it means we've got more work to do. But it made me feel really good that they had ribitol, because that's the first example of ribitol in a mammalian glycan. And so I've shown all the enzymes here, and really one of the things I want to get back to is mutations in every single one of these proteins leads to a form of congenital muscular dystrophy. And these are the so-called secondary dystroglycanopathies. We've talked a little bit, various speakers, about the tertiary dystroglycanopathies caused by DPM 1, 2, and 3 that make the DALP-MAN that allow you to transfer that first mannose. We also have a new tertiary dystroglycanopathy, right? Because this is making the substrate, presumably for FKRP or Fakutin, to add ribitol into this structure. With that, I'd like to just thank Jeremy and Osmond, who did the vast majority of this work. None of this would have been possible without the work of Kelly Moorman. And the second story that I showed you today, the post-2015, was all in collaboration with Kevin Campbell's group at Iowa, where Toby drove that project. And I'd like to thank, of course, our funding. I'd be happy to answer any questions.
Thank you, Lance, for the beautiful talk, and congratulations for the papers. So now his talk is open for the discussion.
That's really nice. So the xylosyl transferase only transfers to the CDP ribitol? And so, I mean, just wondering, how do you think that's going to work biosynthetically?
So I don't think CDP ribitol is its natural substrate. I think the problem is it probably needs to be in a phosphodiester linkage for some reason. Because I thought ribitol 5-phosphate would work, honestly, and it absolutely is a terrible substrate. So my guess is by putting that phosphate into a phosphodiester linkage, right, you're reducing the negative charge there quite a bit, and allowing those electrons to move. And my guess is CDP ribitol is just mimicking that, right? And I didn't show you the K-cat of that, but that K-cat's not that impressive. My guess is once we figure out how to get FKRP and Fakutin to add that ribitol, assuming that is the enzyme that's doing it, that will be a very, very good substrate, much better than CDP ribitol.
And then putting the ribitol onto the phosphate, it could be some interesting chemistry.
Very interesting chemistry. So the question is it transferring phospholipid or is it transferring ribitol? So I don't want to talk about the Europeans group, but I know for a fact they think it's transferring phospholipid, and that POMK is more of a chaperone for that process as opposed to the kinase itself, which is what Kevin had proposed.
So there's no chance that there's a ribose in there that gets reduced.
Oh, that's an interesting idea.
You
you
put
could
on
start
ribose 5-phosphate.
Right.
But in fact, then the other enzyme, FKRP, maybe.
Yeah, that's actually a great idea. Is it could reduce it. That's an interesting idea. I don't know. That's an experiment that I'll do next week. Yeah. Actually, I won't do. I'll tell someone to do.
Thank you. Very nice. So does this allow you to find the epitope for the 2H6 and the VIA4 antibodies? Do they recognize the O-Mannos, GLCNAC,
So what?
or do they specifically recognize what is linked to the phosphate?
Right. So in work we haven't published, so we've built a lot of repeating disaccharide now that we figured out if you use B4GAT1, you can actually get that reaction to go. And that, just the repeating disaccharide will bind to 2H6 with very high affinity. So my thought is it's the repeating disaccharide itself.
That will do the laminin and extracellular matrix binding.
It will absolutely bind laminin, it will absolutely bind 2H6. And those are the only two things we've tested.
If you would desialylate alpha-dystroglycan, would it still bind on the laminin overlay assay?
If you desialate alpha-dystroglycan, and this is not work from my group, it's work from Kevin Campbell's group, if anything, it improves binding. It definitely does not hurt, right? But actually, you're getting back to you just because you're the person who asked the question. The reason I was asking you about the transporter is SLIC35A1 came out of a genetic screen looking for defects in alpha-dystroglycan binding 2H6. It's the only other C-using sugar nucleotide. My guess is SLIC35A1. That's our working hypothesis. That is going to be the Golgi transporter for CDP ribitol or CDP ribose.
They use the same transporter.
That's our guess, right? Because most nucleotide sugar transporters care more about the nucleotide than they care about the sugar. Yeah, absolutely.
Kind of two ideas. When our patients get muscle biopsies, histochemistry, Marison there, dystrophin there, they're doing antibodies, aren't they? They're not. There's no way of finding out if there's any glycan abnormalities. And then the second thing is,
right,
in these primal-derived conditions, is an O-glycan analysis positive that often from plasma?
So we came up with a blood test a couple of years ago because we were trying to get away from muscle biopsy, and we've only tested it in mice. So I would love to talk to people that have plasma samples for of confirmed CMD patients. Because if all you're trying to confirm is 2H6, is there a defect in O-Mannosylation? At least in the mouse models, we can do that blind. So Kevin Campbell has all the mouse models, or the vast majority of them. And so he sent us serum samples blinded, and we were able to determine which ones had defects, and actually in several cases where that defect was in the pathway. And so, but we haven't done that in human patients. I don't know the answer, but theoretically that's going to be possible.
It's usually what happens is the patient comes to us with weakness, hypotonia, developmental delay. I mean, is it a dystrophy, a myopathy, mitochondria, many classes. So
right.
If it comes to a muscle biopsy because lactic acid is normal, there are no other small molecule abnormalities, it's like what is the diagnostic algorithm that really works? Because I'm not going to necessarily think of this.
Right. I mean, muscle biopsy is the gold standard in the field. Right? And that's all antibody staining. That's definitely the gold standard, because no matter where those mutations of all those different genes, they all result in a loss of 2H6 signal in muscle.
Lance, thank you very much for your nice talk. So we're gonna move on to the next talk. The next talks will be Taroh Kinoshita. As a Japanese, when I was a grad student, he was already a famous professor. So it's very awkward for me to call him Taroh, but I just take this opportunity to call Taroh. So his talk title would be where? Oh, he's gonna use.
No, this probably doesn't work well.
The title of his talk is Molecular Basis of Congenital deficiencies of GPI anchors.
Okay.
So Kinoshita Sensei, please.
Okay, thank you, Tadashi. I would like to thank HUD for inviting me to this exciting meeting, symposium. I work in Osaka University, and I have been working with GPI anchor for 25 years. So today I'm going to talk about the GPI anchor deficiency. And GPI anchor is the one form of the glycoconjugate, conjugate, and among others. This is actually the glycolipid and acts as the membrane anchor of the many proteins. Right? GPI stands for the glycosyl phosphatidylinositol. If we look more closely about the GPI anchor protein, the GPI consists of glycan and lipid. And lipid is a phosphatidylinositol, a kind of phospholipid. And the glycan consists of the glucosamine, non-N-acetylated glucosamine, 3 mannoses, 2 ethanolamine phosphates. One ethanolamine phosphate is actually the side branch of the glycan, and the other one connects to the carboxy terminus of the protein by the amide bond. So the protein part itself is not inserted into the membrane, but there's the 2 carbohydrate chains of the GPI insert to the outer surface of the plasma membrane. And in human bodies, is more than 150 various different proteins are GPI anchor proteins. And they include enzymes, receptors, adhesion molecules, complement regulators, and the prion proteins also the GPI anchor. And since those 150 proteins depend upon the GPI anchor for their cell surface expression, the GPI is essential for the embryogenesis, neurogenesis, immune system, and fertilization. And this shows that how GPI anchor proteins are generated in the cell and expressed on the cell surface. They are on the cell surface, but the biosynthesis occurs in the endoplasmic reticulum. The protein part and the glycolipid part are separately synthesized on the ER membrane. And proteins that are to be GPI anchored has at the C terminus, carboxy terminus, the GPI anchor attachment signal peptide. And the glycolipid biosynthesis occurs from the phosphatidylinositol. This is actually the stepwise addition of the monosaccharide, fatty acid here, and the addition of ethanolamine phosphate and the mannoses. So 11-step reaction makes the GPI anchor precursor. And then there is an enzyme called the GPI transamidase. This enzyme recognizes the GPI attachment signal peptide, replaces it with the pre-assembled GPI anchor, generating the nascent GPI anchor proteins. And for these steps, more than 20 genes are involved. So the defect in one of them causes the decreased synthesis of the anchor. So some proteins don't get the anchor. So you get the decreased cell surface expression of the GPI anchor protein. That causes the problem. And the structure of the GPI moiety in the nascent protein is still immature. Then on the way of the transport to the cell surface, and the GPI structure are remodeled to become the mature structure. Both the glycan part and lipid part are remodeled. For those maturation reactions, at least 4 genes are involved, and those genes are called P-gap genes for the post-GPI anchor attachment to protein 1, 5, 3, and 2. If those genes are defective, GP anchor proteins with abnormal structures are expressed on the cell surface. So that causes also the problem. Okay. And if the GPI biosynthesis is abnormal, what would happen is the complete deficiency of the GPI biosynthesis causes embryonic lethality. And this is shown by the pigway gene knockout mouse. And only the incomplete deficiency, partial deficiency causes the diseases. And there are two modes of the incomplete deficiencies that cause the diseases. One disease is the paroxysmal nocturnal hemoglobinuria, or the PNH. The other is the inherited GPI deficiencies. So PNH is an acquired disease of mainly adults. And this is deficiency, GPI deficiency is caused by the somatic mutation in pigway gene. The inherited GPI deficiency, IGD, is the inherited disease of kids. And the deficiency is caused by the hypomorphic mutations in pig or PGAP genes. Right? So the next, I will talk about those two genes. So first is PNH. Before that, I forgot. So this is a summary of the discovery of the CDG genes, and reviewed by Dr. Matthijs. And in this history, the GP anchor study had two initiative roles. Right? It's the somatic mutation in pigway gene was found in 1993. We reported 20 years ago. So that is actually the first report of the identification of the mutation in the glycosylation deficiency. And in 2010 is the report on the mutation in the PIGV gene. This is the first report of the success of the whole exome sequencing in identifying the causative or responsible mutation in the CDG. So what is the PNH? Is an acquired disease. The number of patients about 1 in 100,000 population. So I would expect about 2,000 PNH patients in the States. And this is caused by the somatic mutation of the pigway gene, which is an X-linked gene. And the somatic mutation occurs in the hematopoietic stem cell. So only the blood cells are affected. And the mutations are usually now mutations, very severe, almost complete deficiency, but only in the blood cells. And that caused the deficiency of GP anchor proteins expression, including the CD59 and decay accelerating factor. They are the complement inhibitors, protect host cell, your own cells, from the destructive action of complement. So the pigway gene is on the X chromosome. And if you think about the hematopoietic stem cell, the somatic cell, in the male cell has only one X chromosome and only one pigway. So if the somatic mutation occurs in this one hit causes immediately the GP anchor deficiency. And the female stem cell is they have the two X chromosomes, but one of them is inactivated during the embryogenesis to have the similar expression level as the male cells. So in the stem cell, only one pigway is active. If the somatic mutation hit this active gene, again, this one hit causes GPA deficiency. So in this case, the male to female ratio of the patient and the equal 1-to-1. Okay. And pigway mutation causes the abnormal clone of the stem cell, and that clone generates large amount of affected blood cells, deficient in the CD59, DAF, and other GPNK proteins. And those cells are very, very sensitive to own complement. So it's the major symptom of the patient is complement-mediated hemolysis. And also the complement-mediated venous thrombosis and bone marrow failure. Those are three major symptoms. And if you stain the red blood cells, all the normal red blood cells express DAF and CD59. These are the background. So they're very well protected from the complement. But if you stain the patient red cells, patient has both normal red cells and affected red cell. It's a clonal, big clone. And those clonal affected cells are deficient in both CD59 and decay accelerating factor. They are not protected from the complement. So when the patients get the infection, for example, the complement is activated, then this clonal cells are hemolyzed by the activated self-complement all at once, and gets the hemolytic attack. That's the cause is paroxysmal hemoglobinuria. Okay? So this is the one form of the GPA deficiency, the acquired form. And the second one is the inherited kid diseases. The audience is more interested in this part. Okay? And so this deficiency is caused by the hypomorphic mutations in the PIG or PGAP genes. Right? So to date, the pathogenic mutations are found in the 13 PIG and PGAP genes. And the total number of affected kids reported to date is nearly 100 the cases. And PIG-V mutation is initially found by the whole exome sequencing. The disorder caused by those mutations, the hyperphosphatasia, high level of alkaline phosphatase in serum. Hyperphosphatasia, the mental retardation syndrome. And then the mutation in the PIG-O, PGAP3, PGAP2, and PIG-W all causes a similar, almost the same disorder. So it's called the HPMRS 2, 3, 4, and 5. And the mutations in the PIG-N doesn't cause the hyperphosphatasia, but otherwise very similar disorder. And it is called as multiple congenital anomalies hypotonia seizures, MCA. This is an OMI naming. I don't like this. It's too complicated. And mutations in the PIG-A, this is not somatic. It's the inherited hypomorphic mutations cause a similar disorder, and the mutations in the PIG-T is similar disorders. Okay. And the hyperphosphatasia mental retardation syndrome, also called the Mabry syndrome, was first reported by Dr. Mabry in 1970. And this syndrome is revisited recently by the German and the Canadian research groups. And those are the affected kids. There's the hyperphosphatasia, high serum level alkaline phosphatase with the neurologic deficit. And mostly they have the seizures. And also there's the brachytelephalangy is the dysmorphism of the terminal digit, and they usually associated with the hypoplastic nails. And the severely affected individuals have this anomaly in some organs, and the heart, kidney, and anorectum. This is the symptoms. Then there's the Peter Kravitz and his colleagues in Germany did the whole exome sequence of the Mabry syndrome and identified the base substitutions. It's the biallelic, the amino acid changes. And PIG-V is encodes the mannose transferase. It is transferred the second mannose to the GPI intermediate. And we contributed to this work by the functional study. So we tested whether those mutations caused a decrease in the PIG-V function. And our system is we used we have the PIG-V defective Chinese hamster ovary cells, and those cells don't express GP anchor proteins, and very low, almost negative, and the empty vector transfectant here. And if you transfect wild type PIG-V cDNA, you see the recovery of the GP anchor protein expression. And this is one of the patient mutant cDNA. Recovery is very, very small. And this is if you use the very strong promoter-driven vector, you see the better recovery, but still this is the subnormal. And if you do the Western blotting, this mutant is very, very small amount. So our conclusion about this mutation is the mutant protein is unstable and the total function is decreased. Okay, total activity is decreased. And as expected from that decreased function is on the patient cell surface level of the GP anchor protein is the subnormal, is the decreased. Okay, and we studied the mechanism of the hyperphosphatemia, and we used PIG-V deficient CHO cells, and we used the placental alkaline phosphatase as a model alkaline phosphatase. If we transfect PLAP cDNA into the wild type cell, you see the high level surface expression of alkaline phosphatase, but if the transfected into the mutant cell, it only minor expression on the cell surface. If you measure the enzyme activity from the wild type cell, most of the activity is cell associated, only small amount of on the culture supernatant secreted. But from the mutant cell, most of the activity are secreted and only small amount are cell associated. So the alkaline phosphatase is secreted from the PIG-V defective cells. And I don't show this the data, but our model is like this. In wild type cells, the alkaline phosphatase, the precursor has a C-terminal GPI attachment signal sequence. Transaminase cleaves the peptide bond between omega and omega plus 1 and generates substrate enzyme intermediate. And this intermediate is mediated by the thioester linkage between the C-terminal and the catalytic system. And that thioester bond is attacked by the terminal amino group of the ethanolamine phosphate of the GPI. Then the anchoring completes. But in the PIG-V mutant, the transaminase can make a fast reaction and make a thioester linked intermediate. But the GPI is truncated due to the PIG-V deficiency. So there is no terminal amino group to attack this one. So there is no GPI attachment. But the thioester linkage is eventually hydrolyzed and the soluble form of the enzyme is generated. This is secreted to the outside. So this is the mechanism of the hyperphosphatasia. Okay, this the development of the therapeutic measures for IGD has been very slow, but I can give you the one example is a promising treatment and to control the seizures in the IGD. It's the administration of the pyridoxine that has some the positive effect. In the again, this alkaline phosphatase on the neuronal cell surface, and there is a vitamin B6 is the pyridoxal phosphate is the active form. It's in circulation, but the pyridoxal phosphate cannot go into the cells inside the cell because of the phosphate. And the phosphate has to be removed by the alkaline phosphatase at the cell surface. Then pyridoxal can transmembrane incorporation, and then pyridoxal is rephosphorylated by the pyridoxal kinase in the cell. Now you have the active vitamin B6, which can act as a coenzyme of the gamma aminobutyric acid GABA synthetic enzyme. And GABA is important neurotransmitter in regulation of the neuronal circuit. So this is a normal situation that in case of the GPA deficiency, the surface expression of the alkaline phosphatase is decreased. So this the dephosphorylation of the pyridoxal phosphate is inefficient. So this the incorporation of the pyridoxal into the cell is low. So this causes insufficient vitamin B6 in the cell, and this leads to the decreased GABA synthesis, and that leads to the seizures. So this is I think this is the one mechanism of the seizures in the IGD. So then the idea is the administration of the pyridoxine. Pyridoxine is approved as the B6 supplement. So we can give the pyridoxine to the patient, and pyridoxine can go into the cell, and then can be phosphorylated by the pyridoxal kinase, and then pyridoxine phosphate can be oxidized by the oxidase to the pyridoxal phosphate, and then that helps the GABA synthesis and leads to the amelioration of the seizures. So this treatment has been successful in the several individuals. So this is the promising treatment. Okay, so my colleague Yoshiko Murakami, who used to be the pediatrician, is now the basic scientist. She generated the IGD website. This is in the Japanese page, but we also have the English pages. So please visit the site. Okay, and so this is a picture from the first meeting of the IGD Patient and Family Association held in Osaka last October. And this is the Yoshiko Murakami. And the 5 families, the 7 affected individuals, is the case came, and and those are the doctors seeing those kids. And so we had very exciting time there, and I hope eventually the association will be incorporated and getting more active. Okay, so the major person who contributed to those work is the Yoshiko Murakami. She's there, and Dr. Inoue. He's the medical geneticist. This person, okay, and the others, and Peter Kravitz and Berlin groups. Okay, thank you very much.
Thank you very much, Kinoshita Sensei, for his beautiful work. Maybe a couple of questions from the audience. No? No, if not, maybe we
can talk to later.
Thank you very much, Kinoshita Sensei. So we move on to the third speakers. Can I? Sorry. So the third speaker of this session is by Dr. Eva Morava. The title of her talk is Using D-galactose as a food supplement in phosphoglucomutase 1 deficiency, please.
It's an honor to be here and talk about this really simple and exciting treatment in a relatively new disorder, PGM1 deficiency. I said relatively new, and actually it's not true, because in 1978 already it has been described as a glycogen storage disorder with myopathic features, and later in 2008 by a French group redefined as glycogen storage type 14. And then in a few years, we redefined it as a CDG. Isn't that exciting? Actually, we found this gene as a possible target in 2012 when we did exome sequencing in patients with unknown CDGs. The biochemistry is still really challenging. And just very briefly, let me take you through the pathways. So PGM1 is really essential in the interconversion of glucose 6-phosphate and glucose 1-phosphate. And if you look at this graph, you will see that actually this enzyme has a key role in glycolysis, in glycogenesis, and glycogenolysis as well. So it has connections with different pathways, also proximity to the galactose pathway, and theoretically, of course, with glycosylation. And that's what we see, actually. If you have a defect in this enzyme, we don't only see the muscle symptoms, hypotonia, CK elevations, and the hypoglycemia, what we regularly see in GSDs, but we also see a glycosylation disorder. So how do the patients present? Actually, they have a recognizable clinical presentation. They usually have a flat profile. Many times they have cleft palate and midline cleft palate or Pierre-Robin malformation. Some of the patients have very subtle midline anomalies like a bifid uvula. And in general, they have a really beautiful face and fully normal intelligence. So what's really unexpected in a GSD, of course, to see all these CDG features, including abnormal coagulation. Patients have bleeding disorder and thrombotic events. They have diverse endocrine abnormalities from growth failure to hypothyroidism, to late puberty, and severe liver function abnormalities, additionally to growth delay and hypoglycemia, which can be present in both GSDs and glycosylation disorders. The actual biochemical abnormality is quite interesting. It's we call it a mixed profile. What you see that actually there is some of the normal transferring structure, but we also see truncated glycans. And actually, in the truncated glycans, it's really obvious that galactose is decreased. And additional to that, you also see some desilo pattern, so full missing glycans. So you see pictures of both type 1 and type 2 glycosylation abnormalities. when you stain the cells, the patient's fibroblasts with ICAM staining, you see a significant decrease or a total absence of staining of cell surface ICAM. So let's go back to the biochemistry. This is still really challenging and we don't really understand it. When we actually published a larger series of patients together with Hud and Markert from Germany, we did some measurements in fibroblasts and we found quite surprising results actually. So what we measured is an actually elevated GLUC1 phosphate. So if you think about the pathway, of course, PGM1 is bidirectional, but the preferential direction is to liberate glucose for glycolysis in hypoglycemic or fasting episodes. So it seems like the glucose 1-phosphate is elevated in defective fibroblasts, but also the galactose 1-phosphate was elevated, although in a less severe degree. And also UDP glucose and UDP galactose was abnormal, and this ratio was elevated compared to the normal ratio, and that ratio is close to 1 in healthy cells because the epimerase enzyme is also bidirectional. So it seemed like there is maybe an abnormal or unbalanced flux towards protein glycosylation. You can only hypothesize that based on this abnormal ratio and based on the decreased galactosylation we actually measure by MS in the patients. So that brought the idea, maybe we can add some more galactose to the cells, feed the cells with galactose, and let's see if this abnormal flux normalize. So what happened there, it's very interesting to see that even in 6 days, untreated cells with this moderate galactose concentration starts to show revitalization of glycosylation. And if you use a little higher dose, you see this effect much more prominent. And if you keep the cells in culture for a longer time, you see a significant upregulation in the presence of galactose. And we picked actually this dosage for our clinical trials because this is actually still close to a normal osmotic situation when you add it to your normal glucose intake. The 2 millimolar hasn't been tried in clinical experiments yet. This is just showing you the ICAM expression with with the 0.75 galactose in one of the patients, showing increase in ICAM expression. So how about galactose? I mean, we eat galactose every day. It cannot be so dangerous. That's our luck because it has quick absorption. It's much quicker to metabolize than if we give lactose because we don't have to cut it. There are no side effects. Galactose has been used for people who are for example, bicycle maniacs to offer extra energy during bicycle races. And we know that even a dose of 50 grams in one dose that has no effect on health. It doesn't increase galactitol excretion and it has no side effects. And it's widely used as a probioticum. People use it to feel more energized and freely available on the market. Also, it's supposed to have an immune stimulant effect. I'm not sure if you guys are all on galactose, but maybe this is the time to think about it. So we already did some pre-trials in our initial patients, and they tolerated galactose quite well. So we knew that about 1 gram per kilo is accepted without like diarrhea or any side effects. And what we tried to do is an observational trial and offered the possibility to try galactose in different dosage for a short period. So we did 6 weeks of 0.5 gram per kilo per day, and then another 6 weeks with 1 gram per kilo per day, and then we tried in 1.5 as well, but we never gave more than 50 grams per kilo, which would be about 5 glasses of milk in galactose equivalent, so it's not so crazy. We did all kind of blood tests. We followed the coagulation and the anticoagulation factors because that's the most worrisome in our patients with PGM1 deficiency in terms of long-term survival, endocrine labs, some of the biochemical parameters, and safety stuff, including galactitol and GAL1P, which is actually measurably elevated in patients. So if you actually screen them for galactosemia, they would pop up as abnormal by the newborn evaluation. Okay, so we also, of course, clinically tested the patients during the observational trial, and we followed the heart and the liver function quite closely if we see any negative effect. So far, we had 4 or 9 patients in the observational study with 4 males and 5 females, different age groups, different mutations, and all with a significant decrease in enzyme activity measured in fibroblasts. One of the patients already included in the Tegmeyer paper on lactose trial. In that paper, we reported that there was not much improvement on lactose on that patient in a 6-month trial, actually. And from our 9 patients, 2 patients were pretty non-compliant, and one of the patients unfortunately died also due to cardiomyopathy. Since then, we have 3 more patients on trial right now. So we get this report sheet with all the data. We followed the transferrin changes. As you see, actually in 6 weeks, you already see some improvement. What you see actually disappearance of the truncated glycans and the actual D-sialo in A-sialo, so the missing glycans, they remain on galactose therapy, and the truncated glycans slowly disappear, although not fully. The patient's transferring wouldn't get normal during the trial. But the GAL1P normalizes in a few weeks, which is also exciting because you have an elevated GAL1P, you give galactose, and it goes back to normal. Wow. One of the best parts of the study that the coagulation results, they get close to normal already in 6 weeks. And this is 2 patients, different factors, and you see how the factors from like severely decreased, like 10%, go up to 50% in 6 weeks on only 0.5 gram per kilo, and then normalize pretty much for the 12th week. And you will see that I don't have graphs for all the patients for all the abnormalities because some patients present hormone abnormalities, others with coagulation abnormalities. We don't have really a patient who would present with all abnormal parameters. That's just another parameter. We followed tyrosine binding globulin. Hypothyroidism was present in some of the patients, clinically also, not just biochemically. So here you see the actual 3rd data point as well, and steady improvement in these parameters. Now, this is a chaotic graph, but I want to show you how different the patient's levels are. So this is ALT levels, and obviously we want to have our patient's liver functioning well. So we start with somewhat elevated liver function tests, 100, not mind-blowing high, but you see that in the first 6 weeks, they almost all go back to normal level, except for this one, and that's our patient who was on lactose before. And when we switched him from lactose to galactose, obviously that was half of the dosage we were given. And then when we keep on elevating the actual dose to 1 gram per kilo, he starts to go back to normal and finally ends up where the other patients are. So how about this one? This guy was doing quite well at the beginning. You see it was decreasing well, and then he got a severe gastrointestinal infection, a week of diarrhea, and the levels went back to abnormal just within that 6 weeks period. So actually, the body reacts to the absence of the actual treatment compliance very quickly. So to round up, see other levels like AST levels, they sort of significantly decrease. Maybe they don't actually reach the target range, the fully normal. Here is our patient with diarrhea again, misbehaving, but your levels are close to the physiologic normal levels. And this is the patient on lactose. You see, we could get him back to the range as well. And here is some summary of our results so far for you, which is quite promising, because the most important thing is that these patients are only taking a dietary supplement freely available, a sugar. They just mix in their morning cereal. They add a couple of spoonful of galactose to their muesli or what they eat in the morning, and that's enough to completely restore the coagulation abnormalities in 6 weeks. The liver function tests are normalizing or almost normalizing in a few weeks. And there is significant endocrine improvement. There is not total restitution, but there is a growth spurt in all of the patients on galactose, and maybe a little bit slower than the other levels, but the hormones go back to normal as well. The transferrin, it doesn't get back to normal. You see how slowly it gets better, but it still has a tricylo band in this particular patients, and some patients keep their type 1 pattern, although mild, but still abnormal. However, the actual clinical, physical, and coagulation parameters could go completely to normal, even though the transferrin shows us that there is no complete restitution. It seems like 0.5 gram per kilo galactose is really important. It makes an impact, but if I can make a suggestion, I'd say 1 gram per kilo works better and very quick. In 6 weeks, you see normalization of laboratory parameters. And if you stop it for longer than a few days, you will see it clinically and also in the lab results. Maybe just 2 additional remarks. The patient who was on lactose and is reported in the TechMaya paper not reacting to lactose, he remained on lactose for 3 years. And in the 3 years, he got to the point where we can get him now in 6 weeks. So if somebody cannot get his hands on galactose, then you still can start the therapy with lactose, but it will take much more longer to get the same effect. So the bad news, actually the hypoglycemia and the muscle symptoms do not get better on galactose. Not much better. Obviously, you have to follow a frequent feeding complex carbohydrate diet, because this disease is 2 diseases at once. It's a GSD and glycosylation disorder. And the other sad part that Unfortunately, the cardiomyopathy doesn't seem to improve on galactose either. It's either a genetic cardiomyopathy and doesn't react to biochemical intervention, or maybe just galactose not the right way to help the muscle function. Another problem is the compliance problem. When your treatment is only a spoonful of galactose in your morning cereal, maybe it's hard to really take it seriously. And we see that the patients improve and then they just get sloppy and then forget about it. You have to contact the patients. My dietitian is calling them every 3-4 weeks. Please take your galactose because you will get significant bleedings if you're not compliant. I wanted to show a slide with the World CDG conference in Belgium next year, but then I just tell you, so the World CDG conference will be in Leuven in 2017, and thank you for your attention.
Thank you. It was a really nice talk. So
Lynn.
Yeah. So, I mean, galactose, we know at least for the heart muscle, wouldn't be a fuel of choice anyway. So the question is, and we've talked about this before, especially now that uridine is FDA approved, is there any plus to trying galactose and uridine together?
I think that's absolutely thank you for your question. In actual cell experiments, it seems like a UDP would be an absolute beneficial effect on the biochemical parameters. Whether it would help the heart, that's a different question, but there is nothing wrong trying it. So I think that's the next step we need to do.
As a parent of a young child, I know it's always difficult to feed your children sugar. Even though it's sugar, it sometimes has a different taste. I know mannose has a little bit of a bitter and an after flavor. Does galactose have any of those weird flavors that it might be difficult to convince young children to drink or eat?
Mishak, thank you for your question. Galactose tastes quite good. It's not so sweet as sucrose, so it's less it's less sweet. I cannot put it in a different way. No weird taste at all.
Please.
Very nice, very impressive. So when I was in Belgium and saw a young teenage girl, I said, who has PGM1, I said, what is your biggest problem? And she said, puberty. And I'm wondering if she was on the trial, or if anybody was on the trial, if she'd been able to see any changes, since then.
Thank you for the question. So 2 patients on the trial went into puberty. So that's really a nice improvement. Also growth spurt, because these patients really suffer from being short, and which is not so bad if you compare that to coagulation abnormalities and bleeding. But one of my patients couldn't hike because every time he went uphill, he got a nosebleed really covering her face, and it's completely gone just 6 weeks eating a sugar. That's sort of nice great story.
Any other questions? So if not, thank you very much, Eva. And please join me thanking all the speakers on the session.
Well, thanks to everybody, to all the speakers and all the families today. I'm not going to keep you more than a couple of seconds here. We start up, tomorrow morning? I guess we start at 9 o'clock for the talks. We have registration, which you all don't have to worry about. So if you want to come in a little bit early and talk to folks and kick some ideas around, please feel free to. Otherwise, this concludes the meeting for today. And I've been myself personally excited and really thrilled to see all the presentations. You know, a breakthrough structural discovery, multiple sugar therapies. I mean, this is about as good as it gets. Okay? Great. Thanks a lot.